I'm pretty sure the first job I worked out of college was indeed an Elizabeth Holmes fever dream. If you don't know who she is, she's the woman currently serving 11 years in prison for deceiving investors, fraud, essentially, on equipment that already worked on. It was already invented in just a different scale. Let me explain. Elizabeth wanted to create a clinical chemistry benchtop analyzer that would check 200 tests with a drop of capillary blood, preferably from your fingertip. And we already did that. We had that instrument before Elizabeth was born. In fact, this glucose test was launched in 1985. Elizabeth was launched in 1984, but we had tests before this. This was the Abbott Vision system, and I worked on it. And if I didn't have evidence, I would literally think it was Elizabeth Holmes' fever dream. So what I worked on was indeed a benchtop analyzer. It was in small doctors' offices. It could run 20 tests at once in these little boxes. You put a drop of blood via a capillary tube in the top or a little drop via there into the unit, spin, 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 run, 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 and you got a result. Now, it didn't just take one drop of blood for 200 tests, so there's a big difference there. However, the theory is exactly the same. And we had 30-some diagnostic assays. They included immunochemistry assays like thyroxine, which was my product, CRP, glucose, cholesterol, all sorts of lovely stuff that you would want to run in your doctor's office. And you would get results in anywhere from a minute something to 40 minutes. This is her. That's the Abbott Vision. Yes, it looks like it was designed in the late 70s or early 80s because it indeed was designed then. For those of you curious how it worked, it was a centrifuge that had a spectrophotometer in it, and that is exactly what Elizabeth was trying to invent too, except we did it first and actually launched and went on market. These clever little sonically welded polycarbonate boxes contained a reagent and a diluent that would fall or break or release somehow into this little area, and then the spectrophotometer would read that. The scanner would read the test, so it knew it was testing, and the blood would fall through, everything would fall through these little mixing areas. It's exactly what Elizabeth was trying to do. Now, this was a great product, in my opinion. However, Abbott got in trouble with FDA and had a fine levied against them, one of the largest fines ever levied at the time against a medical device company, for their quality system problems, and I was in quality. This was my product. This one didn't get to stay on the market. This one did because it was medically necessary. And then Abbott just decided they were going to phase out the technology. But listen, they were the precursor. If they would have stayed with it, they could have done exactly what Elizabeth Holmes was trying to do. Which is to say that Elizabeth wasn't smart enough to know that we already did it in the 80s. And if she would have researched the diagnostic industry and maybe stayed in Stanford, she would have figured that one out. She could have worked on the already obvious and available research that was done for this system and just made it better. And in fact, someone still could.